only have very limited capacity, so it's only 30% occupancy, and you're not allowed to travel more than 5 kilometers outside your home.
Oh, great. So take care. I mean, it's terrible.
Yeah, it's quite stressful. I think there was a new strain that became quite much more contagious in the UK. So I think that was happening.
Delekar sir, are you ready?
Sorry?
Hello, sir.
Yes, yes. Are you ready?
Yeah, 3. Okay. Can we start, Lean?
Yeah.
Hello, Lean.
Yes, hello. I'll share my screen.
Just wait.
We'll have an introductory program and then we'll move ahead.
Professor Gule, sir, can we start?
Yeah, no problem. Most welcome.
Yeah. So first of all, on behalf of organizing committee, good afternoon to the Indian participants, and in the participant. Okay, we have there is one participant from Korea. So good evening to the Korean participant, and you know that today's speaker is Dr. Lin. She is from UK, and in UK there is morning. So good morning to the Dr. Lin. So it is the beauty of the virtual the platform. So one can connect the different parts of the globe through the Zoom platform. So anyway, we are in the third day of UGC Stride lecture series, which is organized by Department of Chemistry, Shivaji University, Kolhapur. And in this third day session, we have the eminent the speaker, eminent scientist, Dr. Lin Denny. And before that, in this session is to be chaired by Professor A.V. Gule. So I take the couple of minutes to introduce Professor Anil Gule. Presently, he is working as professor in Department of Chemistry, Shivaji University, Kolhapur. Then, as concerned to his the educational credential, he completed his UG and PG from the Pune University, and after that, he joined for the PhD at one of the reputed national university in Taiwan. He has completed his PhD from one of the institute from the Taiwan. Then after that, he completed the postdoc at Taiwan, then South Korea, then United States of America. So he has huge experience about the research endeavors. Then after that, he had been joined as a lecturer at Dr. Baba Sahib Ambedkar Marathwada University. It is one of the state university in Maharashtra, India. And then in 2014 onwards, he is working as professor in Department of Chemistry, Shivaji University, Kolhapur. So along with that, he has also additional responsibilities. Presently, he is also director of international affairs sales of our university. In addition, he is also the coordinator for IPR sale of our university. Then, as concerned to his research credentials, so far he has published more than 100 papers of international repute. Then, having citation more than 2,700 plus. Then, as concerned to his H-index is more than 29. So, so he's really actively engaged in teaching, research, and extension activity of our university. So with this, once again, good afternoon, good morning, and good evening to all the participants. And may I request, Professor Gule, please take the charge and proceed further. Thank you.
Thank you, Professor Delekar, for your kind introduction and good words about about me, and thank you very much. So it's proud privilege of mine to introduce today's speaker, Dr. Lynn Den, and she's presently working as senior lecturer in the Department of Pure and Applied Chemistry from the University of Sheffield, United Kingdom, sorry. And she is a dynamic researcher that is proof from her credentials, like she has completed 7 mega projects, 5 are active, then she has about 67 publications, and the citation index goes to like 1,257. So it's not only that, she's been awarded with an analytical bioscience group. That shows that her contribution to bioscience and biosensing and all these things. She's also been awarded with the Higher Education Academy Award, and apart from that, Travel Grant Award in 2012. Actually, her background, if I give her background, she's completed a bachelor's in analytical sciences from Dublin University, like in 2001. She completed a postdoc from University of Connecticut in the US, like in 2001 and 2002, and then in PhD in physical chemistry from Dublin University in 2004. Presently, like she is having experience of about 10 years and eight months. She is presently working as a lecturer in the University of Sheffield, and from 2017 she is working as a senior lecturer. Prior to that, she was associate researcher, from 2007 to 2009 in Wollongong University in Australia. Then postdoctoral researcher in Biomedical Diagnostics Institute in University of Dublin, Ireland, and associate researcher. So her present research interest is application of analytical detection techniques for chemical and biosensors development. So she's doing extraordinary work, and it's a great pleasure for me to introduce her and listen to her. It's a treat to all the audience to listen to her. She had visited in 2018, I guess, right?
Yeah, 18.
Yeah, so to Shivaji University, and she was one of the speakers in the conference we organized. So it was a great pleasure seeing her here, and we look forward to seeing her frequently visiting India, and particularly in the Shivaji University. So the dais is open for Lynn, please.
Thank you very much for that very kind introduction. And yes, it will be amazing to get back over. It was such an amazing trip last time, and hopefully we will be able to have some of these discussions and collaborations in person in the future. So I'm just going to share my screen to get going. Okay, so can everyone see that now?
Yeah.
So what I was asked to present today, and because there was quite a variety in the audience, and it's always exciting to get people who are senior, but also right at the beginning of really an exciting journey into research, was about how to communicate science, considering your audience, who you're targeting at, the journals, experimental design and ethics. And so I'm going to take you through that. So firstly, I just want to introduce myself to those who don't know me. So my name is Lynn Denny, and I'm based in the University of Strathclyde. That's in Scotland, so right up the top of the UK, in the Technology and Innovation campus. Currently we had snow yesterday, so a little bit colder than I'm sure you all are. And so it's well known for having that cold weather, lots of fatty food and men in kilts. But on the upside, it has some incredibly beautiful scenery. It's well known for very fresh and tasteful seafood, and also for its whiskey, which they celebrate particularly on Burns Night, which was actually on Monday. So that celebrates the poet Robert Burns, where he toasts the haggis. So just a bit of cultural information for you all. So my journey, or where did it start off? Well, as I said, I'm now a senior lecturer at the University of Strathclyde in the Pure and Applied Chemistry department. And before that, I was a research fellow with the National Biophotonics Platform Ireland. So that was in Dublin. And before that, I had the unique opportunity of working on conducting polymers with the Intelligent Polymer Research Institute, which was at the University of Wollongong in Australia, and I was there for 3 years. Prior to that, then, I worked with the Biomedical Diagnostics Group, and that specifically looked at the detection of a variety of different biomarkers, ranging from cancers to other disease markers and cardiac well-being as well. So quite complex mix of multiplexing to be able to detect those markers. And I'd started all of this off at Dublin City University, where I undertook my PhD in electrochemoluminescence and amperometric detection of DNA and DNA damage. So it already started to sort of work within those remits of chemistry, but very close to the biological interface. So what the group is now doing is sort of continuing on that expertise and looking at new ways to detect quite highly sensitive and needs to be selective markers. So we're looking at both primarily in biomarkers, so for cardiac disease, pancreatic cancer, and breast cancer, but also looking at chemical detection in terms of forensic drugs. So both looking at the illicit substance itself, but also the metabolite. And the metabolite is important for people who end up in emergency rooms. So you're looking at very, very low concentrations, but where you have to be both sensitive, so you want to know when they're there, but also very, very selective. For the illicit drugs, you don't want to give a treatment for someone when they've not actually taken it. For any type of cancers or cardiac disease, you don't want to start a treatment when it's not needed, but even more importantly, you don't want to not start a treatment if it is needed. So it's about looking at all of these in very complex matrices and being able to do that. So there's a variety of different biology, chemistry within that interface, and actually some physics and engineering as well, where you try and translate that into an actual device. So very much at the sort of interdisciplinary boundaries of research moving forward. And so for that reason, it's actually very important to consider who your audience are at different times. If they're clinicians, there's no point in trying to describe the fundamental chemistry because they don't understand it, and you need to use their terminology. For the same reasons, you wouldn't use a presentation where you showed clinicians your work, what you would do if it was for fundamental chemistry. So there's quite a range of material there that you need to look at. So to achieve these goals, we have to look at new electrochemical materials. So this can be quite fundamental chemistry, characterizing your materials, looking at how they work, seeing how they will interact with one another and interact with possible interferences. And we also need to develop these so that they can be multiplexed for high throughput. So that would be looking at sort of into the area of physics and engineering, where you look at how they will be scaled up in their instrumentation, so they could be produced at a low cost, but also then how they would actually integrate into a device or a patch if it's for a wearable sensor. So we need to think about the different electrodes, how they might be designed. Again, that's looking at my very clear fundamental chemistry, but also how that fundamental chemistry will interact when you start introducing a biological fluid. So there's quite a lot of interactions there in terms of the interface between chemistry and biology. And again, if you're looking at those types of devices, you need the physicists to start giving us information on how the microfluidics are, that you don't want it getting clogged up or blocked. You want to have a nice flow through, but slow enough that you can get your responses, but not too slow that you're taking hours to get those. So there's quite a lot of considerations within that. So based on that research at the moment, I now have 60 publications, 4 book chapters, and hopefully a book that will come out in April. And I've done 45 conference presentations. So this is quite a mixture of different audiences that I've had to put forward my work to. So I hope this will help you understand the concepts and the considerations that I suggest within this presentation. So one of the things that I start off with is this fundamental chemistry. So can we use electrochemoluminescence, which is the main platform that I work on within my laboratory, and find a solution for our systems? So we do a lot of electrochemical sensing, but primarily work on electrochemoluminescence, as well as doing some just basic electrochemical or impedance analysis. So the principles of electrochemoluminescence are, if it interacts with a substrate which has been modified with a sensing molecule, that you then get a production of light. And this can go through 3 processes. So first you have your electrochemical initiation, where you cause your capture molecule or sensing molecule, usually ruthenium, but we have looked at iridium, conducting polymers, and I'll go into that later on in the presentation. So you either excite this through oxidation or reduction. It will then interact with your chemical, which is your target molecule. So that can be a chemical, it can be like illicit drug. And the example I'm going to show here, it's a scopolamine drug. So scopolamine is an illicit substance. It has got legitimate uses, so it can be used in eye drops for sort of numbing your eye if you need eye surgery, or if there is an infection there to try and reduce pain. So it has got legitimate uses. But in more common aspects or illegal aspects, it can also be used in concentrations to actually dope people's drinks, and cause that is associated with things like date rape, and actually theft, where people have been given this drug in their drink, in a spiked drink. They take it and they basically forget what's happening, or they're easily manipulated, and so they can have their credit cards taken and be robbed in that sense. So there's lots of reasons for doing that. So that will be our chemical interaction, so where the drug interacts with your oxidized or reduced form of your sensing material. When it interacts, that interaction then requires, it causes an excited state molecule to be produced. That excited state molecule then decays back to ground state, and that means that you get a luminescence, and that's our electrochemoluminescence. That will be characteristic in terms of color based on that sensing molecule. So for ruthenium, it's normally an orange color, around 6-10 nanometers. For iridium, you can actually have a wider spread. It can go into the blue and green, or you can have osmium, which has a slightly higher excitation with about 785. And that's quite important, because when you move to different biological fluids, particularly things like blood, where if you had an emission at 610, that's going to just be absorbed and scattered by the blood because of its red color. So you need to be able to shift your sensing molecule to outside the transmission window of blood, so that you actually can record that signal. But that's the basics of our electrochemoluminescence. And how you would show that in the electrochemical initiation is that you can have reduction or oxidation, and this just basically determines where your electron moves between the HOMO-LUMO form, which you can see on the CV. And I've shown this in a cartoon, primarily to show that you can explain basic chemical and chemistry concepts. That can be a way for people who are non-scientists to actually see, as well as having fundamental work, which you can show for obviously other scientists and colleagues that would have a greater understanding. So you can actually show the same piece of information in various different ways, which is all dependent upon your audience. And that is very important, particularly for oral presentations, but also in journal presentations, because you need to really target who is going to be reading this and what knowledge or background knowledge they're going to have, so that you don't have it too basic, but also where you don't, they're not going to understand it and be able to take home the message that you want them to take home. So it's different, slightly different ways here of showing the same type of information. And again, we can do that for the second stage, which is our chemical interaction. And this is where we have a heterogeneous electron transfer from our excited state luminophore or sensing molecule. In this system, it is our ruthenium with our target compound. And again, that can be a biological marker, things like serotonin, dopamine, adrenaline, all of them will interact in this way. Amino acids are particularly good at working with ruthenium, but do not interact in any way with iridium. And so that's how you can basically modify your sensing agent to actually interact with the target molecule that you want. And just then you will have your luminescence. So this is where you will have your excitation. And I've just shown on the bottom right-hand side, the slide, where you will actually see what it looks like. So this is a photograph of what has actually been taken from this electrode here when we run our analysis. So that will be exactly how it was. Now this is quite a high concentration because you can actually see it quite readily, and you could take that with a phone. But when you get down to lower concentrations, you need more sensitivity. But again, we're still working towards moving that where if it's completely dark, you still have a phone being able to detect that with your RGB sensing molecules in a camera. Primarily because that will be quite interesting to be able to see even at very low concentration. Visual eye would not be able to. So within that, that's just a taste, and I'm going to go back to that example later on as we go through the process of how I have used that research to be able to put forward and develop a publication, how we've gone through that. But within your research, when you start off, you need to consider what you're trying to achieve. Now, this can change because research inevitably starts telling us things when we do it. It doesn't work out as we expected, or does something completely different, and then we investigate. So this is quite fluid, but you need to have some sort of indication of where you want to go with it. Once you have something and you've investigated, you need to consider how do you want to present it. So in the system that I showed there, did I want to give it to an analytical paper where I want to show the application and that's my main driver? Or is it that I want to show that I've now achieved something fundamental and I want to, although there is an application, more importantly is the design of the compound and the sensing elements and that fundamental theory. So which one do I need? And depending on which aspect you want, what experiments would you need to support that when you start to put together and build your research project that will then go forward for a publication, whether it be as a presentation or a journal publication? Because you need to be able to support both your aims, but also then your subsequent interpretation. So if I went for an analytical paper, I want to show the application, I might consider, well, what is important for an analytical method development? And those will be things like limits of detection, limits of quantification, linear range. And so those would be important. For a fundamental interpretation, you might be looking at, well, I need multiple characterization techniques to show what I'm saying is actually happening. So maybe I need the electrochemistry, so I need fundamental cyclic voltammetry and other amperometric techniques. I might also need SEM. Sorry, I just got muted there, apologies. So you need to look at how you would mute or how you would interpret that and use that interpretation to support your evidence. And that's really important because the conclusions you draw in a publication have to be supported. Now, you can say there is still more research to be done, but you have to support your conclusions as you go forward. And within that experimental, you need to have experiments that are robust. So, you know, how many replicas do you need to show that it is robust? Do you need a wide linear range? And how do you determine that linear range? I've mentioned that I've looked at illicit drugs and their metabolites in different bodily fluids. But actually, the range of concentrations for those different biological fluids is quite different. You could have quite a high concentration in blood, but you would have a much lower concentration in things like urine and saliva. The same aspect if you look at cancer biomarkers, so something like pancreatic cancer, you can have different biomarkers, but obviously there's going to be much higher concentration in the cysts in the pancreas compared to those circulating tumors, and it's the circulating biomarkers. But the circulating biomarkers are the ones that you would want to be able to detect for a screening method to minimize someone having to undergo surgery to be able to determine if they have pancreatic cancer or not. So you're trying to limit the invasive procedures on the patient. And by doing that, you need to look at a very, very different range of concentrations compared to what you would look at if you were looking at analysing samples taken from biopsies. So you need to consider that your linear range is not only linear and wide, but also that it's appropriate for your technique. There's no point running it and getting incredibly nice results for a really high concentration that in no way helps with your actual application. One of the important things then is, do you need to validate against another technique? So this can be both with the fundamental analysis. If, for example, I take the ECL example there, we could say that it creates a luminescence, and that luminescence is based on our sensing molecule from the electrochemiluminescence. However, if I had to prove that, I would have to look at both our UV and spectroscopic data, but also looking at photoluminescence and other methods of trying to prove that the luminescence is in fact due to that ruthenium molecule and not due to something else in the system. You might also want to look at things like transient absorption spectroscopy to show where exactly that electron is moving from, or high speed electrochemistry to be able to show exactly what is happening for those or different stages of the electrochemical process. So you might want to look at multiple things. If you look at trying to make new molecules, you'll have a huge variety of different techniques that you might want to consider to prove that the chemical you make is it, and then it works as you say. So that might include spectroscopy, but also things like NMR quite regularly used, mass spec, as well as things like SEM. So you have quite a large variety of things, and you need to determine which ones are important to be able to put forward so that you know you will support your conclusions, and that the reviewers that go through it will go, yes, that does prove and support what you're saying there. Again, would you need real samples to illustrate the feasibility of your approach? If we think of many of the journals that look at that interface with biological materials, you have to consider the fact that they want to be able to show that it's feasible and viable in real samples, as opposed to it can just work in the laboratory, but the minute you use human serum or blood, it's not going to work because there's so many interferences. And what is the field norm? So is it commonplace that they would use simulated samples? Is it commonplace that they would use real samples like urine? And how often do they use them? How many? And what's the typical sample base that they would need to prove that? For things like cancer diagnostics, you might want to say you'd look at getting a biobank to send you maybe 100 odd samples from different individuals, to be able to test it to see the natural variation in individuals, because that natural variation will cause issues for your analysis. So you need to be able to show that, and also show that it's statistically valid by looking at the sensitivity and selectivity over a much wider range of samples with diverse compositions. And if you do need those type of samples from biobanks, so that you have to consider ethics. So ethics can be actually incredibly wide. It can be, are you getting any personal information from the sample that you have? Will you anonymize it? How are you going to go about doing that? So there's quite a lot of things that we'll consider in that, and I'll go on to that a little bit later on in the talk. So if I look at how I built in a methodology or experimental in our design, I'll just go back to our example of ECL. So our aim was to try and develop an ECL sensor for rapid and portable detection. So I've already said that it's going to be for an application as opposed to the fundamental concept. So we're already moving towards a particular journal. So in our setup, we want to use screen printed electrodes because that shows that we're moving towards that portable detection, and how we would modify it. And then we want to pick model compounds to illustrate that this is going to work, and it will work in multiple matrices. So the first thing that we looked at was, again, because it's going towards an analytical paper, would be the linear range. So we looked at both atropine, which is structurally similar compound, and our scopolamine. And you can see they've slightly different, very, very minor, but very slightly different linear ranges. But both are appropriate for illicit activities, because you have to have a concentration above 8 or 9 micromolar for it to actually have an effect on an individual. So where you would have spiking drinks were lower than what we would expect, which is where you want to be. You want to detect lower than what you want to do, but also higher. So the range normally for people with getting spiked and actually having an effect on that individual are normally between, so the minimum amount is about 8, but actually the range that is normally spiked will be between 20 to 40 micromolar within the solution. So we're right in that, we're covering that range that we could say, yes, we're in that range and could detect that, and it's appropriate for this example. We then also showed things like our sensor reproducibility and repeatability. So if you're going to have a portable sensor, you have to show that this will happen no matter what electrode you use. It's going to be reproducible and repeatable every time you use it. It's not going to change every time you open up a new electrode batch and suddenly you have a completely different response and a different limit of detection. So you need to look at these different things. And again, within this, you can see how we've condensed a lot of that information into one figure. So we have our ECL been shown here. We have our linear range, which includes our limit of detection. We have the reproducibility by adding our error bars, and we also have the molecule that we're detecting. So within this, you're showing all the different aspects, and you can discuss that, but you're being concise within your imagery while conveying all that important information. That's not always possible for things like characterization, where you might have NMR spectroscopy, and it will get confusing to try and add them all into one figure. So you have to balance how you present that data between what is concise, but also what's clear, because the reader has to be clear on what they're actually reading and seeing. So part of this was, again, a rapid but also a portable detection system. So we needed to assess the sensor's capability to detect in multiple complex matrices. So again, we knew that if we were to show this within the journals that we're going for, we would need to show that it worked in these type of complex matrices. So we chose a variety. We looked at drink samples because that's where they would be spiked in. We looked at herbal material because there is now a trend where you could have drugs being sprayed on plant material and then smoked in the same way that you would, or where it was traditionally cannabis would be smoked. And we're also looking at biological fluids. And the reason for biological fluids, again, going back to your drink samples, is where if they were admitted to an emergency room, you would be able to diagnose that, yes, they had in fact been, they had their drink spiked, and we can tell this and therefore move forward with their treatment. So it would help both the patient or the victim, but also the doctors and nurses be able to actually start treatment and understand what's there and how they can go about, so improving their ability to move more quickly towards treatment of that. So again, this is examples of where we spiked our drinks. So we looked at Coca-Cola. So why did we choose Coca-Cola? And the reason behind this is that basically it's a mixer for a vast majority of different drinks. But because of its dark color, it actually causes a huge amount of problems, because how they would usually screen for spiked drinks would be things like IR and ramen. But with that dark color, they're not possible, because that actually negates being able to see any of the individual molecules present in the solution. It also causes problems for presumptive tests. So their presumptive tests are basically color tests that forensic scientists or police use to determine if there's an illicit substance present. And I'm sure you've all seen this on telly, on either border security programs or on just normal crime series, where they think, oh, something has been spiked with heroin or cocaine, and to test whether that is it, they put a small amount of powder into a little pouch that contains liquid, burst the capsule, and then when that interacts, it changes color, and that color change will tell you, indicate that there is heroin or cocaine present. But if you have things like Coke and you added Coke into that sample, then you're not going to see that color change, because the dark color of the Coke causes a problem. So that's why we chose Coca-Cola. We went for a very difficult substrate that would be causing problems against other techniques. So by choosing this, we have then started to look at how we would assess our technique against other techniques that are used. So that adds to the experimental design by also showing what is novel about this approach and what's going to be significant to the community going forward. And they're very important points to consider when you're trying to put together publications, because the journals want to know that there is a high novelty and high significance. So they want a high impact of the work going into their journals. And so that's why they will choose to pick things that show what that novelty is. So you're supporting what you're going to confirm and move forward with. So again, we also looked at herbal material, not as big of a complex issue, but what this does show is that the same system will work on different types of samples. So again, we're illustrating its versatility, and so therefore feasibility if it was to be moved out and into production. And again, I mentioned biological fluids. And so the important point about biological fluids is the fact that they're incredibly complex. So if you think of serum, you would have to have gotten, first of all, ethics. So what we did to get around the ethics is we went for a human pooled serum. This is a nice start off. It's pooled, so that means there's no information that we can get from that sample that would be specific to an individual, because it's pooled from many, many individuals. So that means that you don't have to consider as much about the ethical considerations that you would normally have to. But it is important that you consider that if you move, the next step would be to individual and a large array of individual serum samples and then blood samples. So what we can see here is that the serum, because it is complex, it does give a background response. Now, this doesn't mean the end of your work. What this means is, what are you trying to show? Can you detect this, and can you detect it over the range of values that you deem appropriate? And within this, because we are getting a background response, we have to put a threshold. So we would say we can see something below this, but if it doesn't go above that, we cannot be confident that there is scopolamine or atropine present. And this is important to show that you know where the limitations of your work are. It doesn't mean that there's anything wrong with it, but just what are the limitations for that? And again, we use artificial saliva rather than real saliva, purely because trying to get the volume needed would have been too awkward in that. So there is also issues when you put forward these about what is actually practical. Trying to get multiple or large volumes of saliva can be very awkward. So again, going on an alternative to prove this before you move on to further analysis is very important. And the other thing that we looked at then was sweat. So again, we had artificial sweat rather than having someone run on a treadmill while we run this. So that was very important to show. And again, we had to look at what possible interferences will be in sweat and what range of concentrations. So we did actually look at the amount of drug that will be excreted through sweat. So we were still in that range, which was good. If we had had a different range, we would have to modify our data set and modify our analysis to be appropriate for the amount that will be seen in sweat, because doing it at a much higher concentration wouldn't be appropriate, and therefore will be commented on probably by the reviewers when they go through this, but also to say, well, hold on a sec, this isn't going to work, it's not feasible. So you need to consider these. And remember, when reviewers come back with comments, primarily they're there to help you with your research, even when they're not saying overall to be accepted. It can help you improve your improve your research, improve your project, and know where the limitations and the gaps in your research are. So a really good review or a good reviewer will give you constructive feedback to be able to see, well, have you done it well? But where you haven't done it well, how can you improve it? What should you consider? So I think it should be looked at as quite a positive experience. Now, that's not always the case, but the vast majority are. They're there to help you improve it, because they're scientists themselves and they know when they put in work, the same thing might happen. So they're looking at trying to improve the end products. So when you go through that, hopefully the publication will be better because of those reviews. Now, the other things that you might need to do are forming your conclusion. So when you have all that data, you have the response on that, they have to be realistic. You can't say you've solved all the problems of the world when you haven't. You need to consider, well, it goes this far and no further, and we need more work to get it to the point where it could be in production. So you should address the limitations of your experimentation. It may be that we've looked at it well, it shows really good promise and potential, but we need to look at it with that much larger sample set to really show its validity for this approach. In terms of a fundamental paper, it may be this is as much as we can do with the current instrumentation that we have. We would like to look at, maybe more NMR or TEM rather than just SEM, but these aren't available. So, addressing the limitations are not a bad thing, but you need to be able to say them and be honest about them. And that will actually strengthen your case for a publication, because it shows that you are considering what are the limitations, and so the conclusions you are drawing are appropriate. There are also, you can use that to say there are future avenues of research. We know that there's an issue here, but we're going to look at it in the future, and here are our future plans. And that's important, particularly if it's something like a proof of concept. It's only going to prove that this might work. There's a lot more work that can be done. Or if you're getting an end product, this is going to be our model complex, but we hope that there will be a large family of these that we will be able to utilize. So, the more work that you can do and showing that there will be future work can be very important. For the example that I gave, you will have noticed for the scopolamine and atropine that they had almost identical ECL spectra. So they oxidized at the exact same potential because they were both working with ruthenium, and that is a problem. So we know that we wouldn't get specificity. We'd see that they were there and we could get it very selective, but we couldn't tell the difference between those two compounds. So that is a limitation of our work. And how might we get over that? Well, we could exploit the differences in how the two compounds react to pH. So we can see that we can have atropine and scopolamine, and there are very differences, particularly at pH 6, where we would see scopolamine but not atropine, and again at pH 11, where our atropine concentration will be seen in much more higher significance compared to our scopolamine. So by utilizing those pHs, we could start to see differences and be able therefore to give information on the specificity which is present and at what concentration. The other thing that we could look at is utilizing different lumiphores. So we looked at ruthenium, and that is a very common ECL lumiphore. But if we looked at something else, we might be able to get specificity, and we looked at some iridium complexes, and we're still actually looking at this, so it's still very much work in progress. Because we want to move away from the illicit drugs and utilize these model iridium complexes for biomarker detection, where we know we're going to have very structurally similar proteins, but one will show that cancer is present and one won't. So we need to have that really good specificity. So this work, although started with the illicit substances, will very much move into the biological marker for cancer detection. And as you can see here, we can have a very clear difference between the scopolamine versus the atropine, actually getting our green response from our iridium complex, but not having any interaction with our atropine. And so these are things that are moving forward. So again, there was a conclusion that we knew we could get a portable proof of concept, a portable device, but there were limitations for what we would want to apply it for. We showed an example with illicit substances, but really our end game will be to try and move into biological matrices, and being aware that we need to improve specificity is essential for any biomarker system. Again, with ethics, as I've mentioned there, you will need to, I think almost every journal now requires that you acknowledge their ethical policy, and that you acknowledge it, tick that you've acknowledged it, and that the work is your own and has abided by the requirements that they've set out. You also have to consider what your national requirements for actually carrying out the work. And these can be very different because there are differences between the UK, the States, Australia, India. They're all very slightly different. And so what you're allowed to use personal information for and how you use biological samples can be very different throughout that. It might mean that if you get in samples, how do you collect that information? So do you store that personal information, but the person doing the analysis doesn't? How might you analyze it or anonymize it? How do you dispose of it? So after a certain amount of time, will you be required to get rid of that information, or can you keep it on indefinitely? And so there are quite a lot of considerations that you need to take into for doing these type of ethical considerations. And again, finally, you have acknowledgements. Again, this is a requirement of all journals that you acknowledge any funder bodies. You might also have contributors. Maybe someone has helped you, but not significantly to be an author, but have contributors, and they need to be given their contribution within that journal for any advice that they give. But it is a requirement that you declare all the funding that you have that has resulted in that publication. Now, it can be multiple types of or multiple funding agents and funding sources. So it's not like it just has to be one. There can be more than one, and it can be different, particularly in collaborations, as I've had with different professors at your university. I mean, they may have funding which has supported them and their researchers, and I have a separate funding for mine and my researchers. And so those can both be taken into account, but both should be acknowledged within any publication that you have. Or again, within any oral presentation, you should always note who has done this work, because a lot of times it's not going to be you individually, but with a team, and it's important to acknowledge that team. So the work that I showed you there, as I said, we looked at that in terms of how we could put it forward for an analytical chemistry journal. And so we utilised our work there and put it forward to AnalChem, because the scope of that journal really tied in with what we were trying to give. And I've just shown what that work looks like now. And you can see that there was multiple people involved. We had Kelly, who's my PhD student, doing a lot of the experimental work, but we also had colleagues from UCLan, Dave Robinson, as well as other colleagues in the Department of Chemistry, Pamela and Math, who all helped us out at different aspects and different points of the project. So it is a collaborative team that has worked for this moving forward. And I've also mentioned that this is the way that we've shown this work and publicized this work through a journal publication, but we've also done a Twitter poster competition. So Kelly was amazing at these type of graphics, and she put forward this for the RSE Twitter poster conference. So this is something that happens every March now. And it's basically you put up a poster on Twitter to share with everyone. You can get questions asked on it, but because it's on Twitter on the public domain, you're going to have a very, very different audience. So there will be some people with scientific knowledge, but there's also going to be people without that scientific knowledge who are you trying to get them interested in science and in doing things, and just wanting that dialogue with the general public. So in that regard, we have to consider, well, it's going to be very different to what you would expect from a standard poster that you put in a conference where everyone would have a scientific background. So in this, what she did quite cleverly is basically move away from that standard poster presentation and actually try to develop it in as a comic strip, which I'm sure, as you can see, it's quite interesting there, and it's available still up on Twitter. You can still find it if you want to look at it in more detail. But I would encourage everyone, if you have a project that you're working on, and as long as there isn't actually a paper that you're ready to get out, because there may be issues with publishing it later on, but get that work at the infancy, at the early stage, put it forward, because the Twitter poster conference actually has a vast majority, so it's worldwide. So you have people coming in from Australia in the morning, then later on from the UK and America, and then going back around. It's run for 24 hours. And you can actually get some really, really interesting and helpful advice from people as they ask questions to you on your poster. So that can be really informative. So by putting your work together in a more relaxed format, you can actually still get some really, really helpful advice as you go through it all. And again, we also have another RSE Analytical Biosciences group. So this is a conference where we're going to run it. So I'm part of the organising committee. So this is completely advertising our event. It's going to be on in March as well. And I've given the link here. And I can put that up on the chat box later on. But this is going to be online, so no longer, because of one of, I suppose, maybe the upsides of COVID, it's the fact that we are doing more things remotely, and that can actually improve how the audience and how many people can access that information that wouldn't be available if it was person to person. So this will have some presentations, and again, you can present it as an oral presentation in a similar way that I'm doing now, but you can also put in posters. So we're going to have posters on that process as well. So that's on the 11th of March. So if you're interested in that, please go ahead. The registration is free. So, I mean, you can get involved in it. You don't need to present anything. You can just see some research. But if you want to, I would encourage everyone to put in either a poster or an oral presentation, because that can be really useful in getting advice, feedback, give you a direction, but also get you more comfortable with presenting as well, which is a key thing within academia now. The next complex, and I'll move a bit quicker on this just to show, I've showed you a very analytical approach, but we also look at fundamental stuff. So one of the things we looked at was the development of new organic ECL molecules. So this was much more fundamental. So I'll show that structure, which is T4BTB. So I'm going to use the abbreviation rather than the full name. So this was a new compound that was being developed, and we wanted to see what its possibilities would be for electrochemiluminescence. So again, much more fundamental. And because of that, then we went for very different journals. So we looked at the Journal of Material Chemistry C and also Faraday Discussions. So again, a mix there for what we would look at. So we tried to show what the compound was, how we would change it. So our structures and NMR to show that how we were changing those arms of that star-shaped structure. Within that, because it was fundamental, we needed to look at the full CV, so looking at our oxidation and reduction potentials across the body of that structure. So where we have our core oxidizing reduced, but also the arms. So we needed to look at all of that to show how it would work. And then we needed to look at its photoluminescence. So we needed to look at both the emission as well as absorption, and also the ECL. So how we would look at that in solution to compare against. So we had to look at very different things to show this is working as we expected and is similar to what we would expect. And you can see that we actually overlapped our ECL with our photoluminescence to try and show that there is the same interactions or the same mechanisms that are causing the ECL production. We then tried to look at our efficiency. So this is a new compound. We wanted to compare against a previous one, which would be DPA, and see how much better, or if it was in fact better. So we looked at quantum yields and ECL quantum yields to show if it was actually better or not. And in fact, we could show that it was the quantum efficiency of our ECL from T4BT was almost 7 times greater than DPA, which is a standard molecule that will be utilized within ECL compounds. So again, using that against what is the current standard to show what the significance of your result is in terms of a fundamental approach. And then we gave all that data. So we looked at our HOMO-LUMO gaps, how it alters, what the emission and efficiency were. And again, then to just image it, for DPA, you would not see any luminescence visually, whereas when we switched on our system, we could actually see, and that is our ECL response down here. So what it would look like when we turned it on and off. And again, so we needed that fundamental electrochemical and spectroscopic data and how we calculated our HOMO and LUMO gaps, but also then showing this is visually what it looks like. So adding that support for the reviewers to see that information. So that's why it's important to look at what type of journal. So did you want things like JACS and ChemCom for more fundamental stuff? For analytical, did you want things like AnalChem or the Analyst? I'm using these as examples there, and by no means the limited ones that you have, just the different ones that you do. If it was electrochemistry, do you want to go for a more fundamental electrochemical journal? So that would be Electrochemia Active. But again, these are just a very small number of the thousands of journals that are out there, and you do need to consider which one, because each of these will have a different format, a different requirements. So you need to be important in that. And within that, you need to consider the scope of these journals. They all give on their main page the scope of the journal, and you need to know, does it align with your work? Because if it doesn't align with your work, then it will get rejected based on the fact that it's out of scope. So considering whether it's in scope or not is incredibly important, because that's the first stage that an editor will look at. Do you have it in a correct format? A lot of these journals have some of them don't require it, but some of them do. So you do need to look at whether it's in that format or not. I will mention that some have word, page or figure and table limits. So things like Electrochemia Acta has a maximum number of figures that you're allowed. So obviously there isn't that for things like AnalChem or JACS. So you would look at, well, I can't put in this amount of data. So do I need to move some to supplementary material, or do I need to look at a different journal because I cannot get the information across within that limit? So these are important considerations. They also have considerations for the style. So a lot of times with figures, whether it's a PEG file or a TEF, and those requirements will be given to you at the journal information page. So you do need to consider how you put forward that information. Another important thing is your cover letter. So all of these require a cover letter, and I would say always look up the editor. It may be the main editor, or it might be one of the associate editors. Who do you want to assess it, and are they in the area that you want? So you can decide, but I would say always address it to that individual. That's going to be the first thing that the editor and the reviewers read. So you need to highlight what you're doing, the aims, why it's important for that, and significant and novel, and also why would it be important for the scope of their journal? Why would their audience like this journal and want to read it? So why would it be important for their readership? To show that it's significant to that journal and that it is in fact in scope. So these are the very important points to put forward in your cover letter. Now, normally I would say only go for a page. It doesn't need to be long, but you do need to be concise with those bits of information to show that. You also have a table of contents image. And again, this is one of the first things that the reviewer and editor will see, but it's also going to be the first thing that anyone who searches for your journal sees. So you want to draw their attention. So you want it to be visually interesting. And it wants to really show what the overall aim of your project is. So a good image can help you get to the review and also through the review, but afterwards you can also say that will attract the readers of your journals, which will hopefully in the long run improve the citations and your standing within the field. So having a good table of contents image, although it is very different for us as scientists, it is important to be able to look at that. And so this is just one of the table of contents image that I showed for this ECL compound. So again, looking at interesting, but also showing that we're trying to distinguish between two different forms of our scopolamine. So as I've mentioned, it's always important to acknowledge everyone that has done. So I've given the whole list of people who are working with me in this, in the group, all my postdocs and PhD students, but also collaborators from Pete Scabara, who worked on the truxurine, new organic molecules at the University of Glasgow, Paul Francis in Deakin University in Australia, Robert Forrester, who is based in Dublin City University in Ireland, Emma O'Reilly, who is in the University of Limerick in Ireland, and Ashton Theakston, who is also from Deakin University. So a huge amount of people to get this work together. And again, it's always more interesting, I think, to do work as a collaborative group, because you will get that feedback while you're going through the process, and everyone can bring something new to the table. And again, the acknowledgements for work. So I've had collaborations with and funding support from Horizon 2020, Deakin University, DCU, UL, and Westchem. And again, all our funding bodies. So a quite snapshot of all the people there. So thank you very much, or Gura Mila Mahagut, as it is in Irish. And thank you very much. And if you have any questions, please feel free to ask them. I'll just stop sharing for the moment.
Thank you very much, Lynn. Thank you for your interesting talk. So it was really impressive, and I think it will be useful for the students, because they are like very keen on understanding like how it's to be put in place. So that's very much, thank you very much. So any questions? It's open to the audience now. I'll just read out some of the questions which are there in the chat box. One of the students, like Ankita Dukhade, she's been asking like how to minimize the effect of interfering substances while analyzing real samples like blood or other fluid.
So there's multiple different ways that you can do this. So one of the things is that you can actually put a mesh right above your electrodes. So it's like a little mesh, and then as you go through the microfluidics, it will get off those much larger proteins and blood cells. So then you're reducing down the amount that's actually been seen by the electrode. Now, if you want to detect some proteins, that's obviously not very good. So you need an alternative. With the ways of doing that is by having some sort of capture molecule or a specific interaction. So this can be by changing that sensing element. So although I showed ruthenium, this is a sort of a standard ECL one, and that would not be useful at all for biological samples, because there's literally so many things in blood that it will react to: amino acids, reactive oxygen species. All of them will interact with ruthenium, so that would not be very good. But you can actually modify that by changing the ligands on that compound, so that can add specificity, or you can move towards a different compound altogether. The other things that you consider can be things like using recognition molecules, so aptamers or antibodies. And again, you could have like an ELISA type system, but still with a lumaphore that is ECL active at the end. So you could still utilize that, but just with a different format. So the other thing then that you can do is trying to create a dual complex. So where you would have, as I showed there, you had like a ruthenium and an iridium, and so they both interact, but only one will interact with the target molecule. So you could get two responses, and that can be like an internal control to show that you're actually interacting and giving the concentration for the molecule that you're trying to find.
Well, thank you very much, Ling. Well, I have one question. Like when you talk about introducing aptamers or like some kind of ligands, but then when you're like immobilizing all those things, the reproducibility again is a question. Like, when you're doing multiple samples, that again adds to the complications. One more thing is they talk about the mesh being put into the flow. So some of the molecules of interest, the analytes, might get trapped onto the protein itself. So there would be loss of concentration, which would be affecting the things also. So there are like a lot of issues we have to address in that.
I think that's one of the things that I meant there by, like, if you wanted to get protein, you wouldn't use a mesh because that will basically block it. But if you wanted to get a much smaller molecule, like something like an amino acid or like dopamine or serotonin, it would be appropriate. So it really does depend on the target molecule that you want to see or want to detect. And aptamers and things like that will be on the structure. Yes, I mean, we try and go for as simple as possible, but sometimes we do need to go a little bit more complex to get the sensitivity.
Simple the best. Yeah. Well, the next question is like, what strategies can be used to improve selectivity of sensors?
So that's pretty much the same there. So we can try and look at different molecules, and the selectivity that's required will be dependent on your application. So for some of the drug ones, it's not really that much of an issue. We can know that one of two things or one of three things will be present. But what I will say, particularly for biological samples, that's where you do have multiple ones where you really need to see it. And there's different things that you can do. You can either have a two-stage approval. So 2 reactions have to occur in order for you to see and get a response, say it's a yes result. Now that can be maybe you have ruthenium and iridium, or ruthenium and like something like T4BT. So only if both react will you say that compound is present.
Kind of sandwich assays and kind of thing, because like 2 binding sites, like polyclonal antibodies and kind of combinations.
Yeah, or even simpler than that, literally 2 assays running simultaneously at the exact same time. So in that, I've looked at just running ruthenium, but you could have maybe like a microelectrode array where maybe there's 2 or 3 different molecules on it, and in order for you to say that your cancer, say CTNI, that's a protein that we're looking at, that it has to react with 2 things, because CTN or CTNT, which is slightly different, will only react with 1. So you have to get both positives in order to be able to say it's definitely present. So it's like simultaneously running 2 reactions at the one time.
Thank you. There's another question, like, what is the ratio of copolymer in T4BT that shows the best performance for your application?
So in this sample that I showed, there wasn't a ratio at all. We literally just have T4BT -4. The ratios or the different N numbers were actually the length of the sort of star shaped, and we found that the T4BT was the best one, but we just used that independently. So there wasn't a ratio there at all. It was literally just that coated on the electrode, and that was it.
Yeah, thank you. There's one more question, like, how can we overcome the life cycle issues seen in the case of redox active material? Like, as we see redox performance get degrade after, like, eventually. So
this is really dependent on where you want to test. So like if I mentioned their pancreatic cancer, so if you get a biopsy, you can actually test it straight away and there isn't an issue there. But because of that degradation, there's no way that you would use that if you were testing in a blood sample. So therefore, something else, because there's so many different things that are going to interact. The time frame will not allow you to see that the minute it's formed. So really, I think you need to consider not only what you're detecting, but also how long it's going to be there for. So the lifetime of that molecule, because some of them, for circulating samples with blood, you're not going to see anything that doesn't have a longer lifetime. Like it will have to have a lifetime that is, probably days.
Okay, thank you, Len, very much. I have one last question. How do you compare the analysis of all these kind of biomolecules compared with electrochemistry and mass spectrometry? Like, how do you, compare them?
So there's different ways of doing this. So what I would normally say is that the electrochemistry has its drawbacks in comparison to mass spec. So a lot of times it's not going to have the sensitivity or specificity that you would have for things like mass spec. However, if you run these, you're going to take like those samples that we ran in with blood, we've even run them in blood, they will take about 30 seconds.
Yeah.
And you can run them directly in that bodily fluid, so there's no analysis. So therefore, you can get something that you can find in an emergency room, or if someone comes straight forward. The mass spec might be if they're, you know, months in and they're looking at work, although it might be more sensitive, it won't give you that instant response and that rapid response that you need. So it won't have the sensitivity that the mass spec have, but it does have that speed to be able to be utilized. So I think in a lot of places there will probably be some sort of combination. One that gives you that triage, if you think of it in an emergency room, triaging your patients, and then the one is actually going through the treatment.
Thank you, Lynn. Thank you for a wonderful answer, and I'm sure all the students and the researchers, they will benefited from all these kind of interactions. Thank you very much, and I hand over the charge to Professor Delaker. Thank you, Delaker, sir.
Thank you very much.
Hi, Lynn. I have a couple of questions.
Yeah.
One is that which property of material is to be considered for efficient biosensing application?
I think that is so broad that I don't know, we'd have to have a full hour just to go through all of that. One of the big things is where it's going to be utilized for biomarking. So is it going to be external to the body or internal to the body? There are a lot of sensing elements that are actually internal. And so things like conducting, or the polymers that are that T4BT, that would not be appropriate at all, because that will degrade and not be good for your body. It will cause more problems than solutions. Whereas externally, that's absolutely fine. Again, for things like ruthenium, they are used for therapeutics, so you can use them within the body, but they are heavy metals, so they build up and your body can't excrete them, so you would need to have them in very, very small concentrations. Again, not an issue if you use things like you know, blood that you've taken, urine, like samples that have come out of the body. So I think they are the biggest issues, because that toxicity and compatibility with the biological sample is a major thing. In terms of developing new compounds, one of the biggest stumbling blocks with developing new compounds is actually having it work and work well in an aqueous environment, because the vast majority of materials that are made are all characterized in organic solvents, and they can be fabulous and they will work absolutely amazingly. And the minute you put them into water, you can get quenching and interactions, and so suddenly they will not work as well. So considering the matrix and how it's characterized is also very important.
Okay.
Well, Lynn, I have one question. Like, you know, I would like to know whether you're working on biotransistor or genetic transistors, kind of, you know, for sensing purpose.
We're not working on transistors at all at the moment.
Because one of my interests is, you know, the conduction between the differential conduction between single-stranded DNA and the double-stranded DNA, which, you know, will be very useful for early detection of most of the disease, because most of the biomarker molecules can be linked to the genes. So, you know, that's what is one of my interests. So I was just looking for somebody who would, you know, collaborate and get the things done.
Yeah, no. We have looked at some of that, and I think one of the big issues really with getting them translated is convincing the clinicians. But yes, there's entirely, I think if you can look at the very differences between DNA and RNA, both single-stranded and double-stranded DNA, that's definitely something. We have obviously looked at some DNA sequences and working on them, but at the moment we're not doing much. But I would be more than happy to collaborate at different points.
Sure. We'll try to work out something on that, because I have some of my collaborators like in Japan and Taiwan. I know they are working on it, but you know, since I've left back to India, we are not able to communicate with them very well, and that's one of the limitations. So we'll be working on, because you know, this is having a good scope for early detection, particularly, you know, that is very important. Thank you very much, Lynn. Yeah.
Thanks very much. And I would say,
Lynn, okay, I have second question.
Yep.
Does it possible to design the multi-analyte sensing in a single component device? It is the need of power.
So, well, I will say that the limitation at the moment for some of these is actually stopping electron transfer between different molecules. So what we are trying to do at the moment is actually create a whole new set of them. So we have a grant currently going in to try and actually address this very issue. But I think there's going to be several different ways that we need to do it, because we would look at the fundamental sensing molecule, but also about how well it is structured in terms of electrode arrays to try and prevent that interaction. Because really you do need to have, for any really impactful and useful, as well as actually going forward to production, it cannot be a single sensing molecule. It has to be multiplex. And I think I'm very much into trying to find out as many different ways of doing this as possible, because I think we need to look at all these different varieties. I don't think there will be one size fits all. I think you will see some diseases that will work a different way, better with different platforms. So I think we need to look at quite a variety of different ways, but a very, very topical question, and one that's very much at the forefront of all research at the moment for biomedical diagnostics.
Okay, thank you. Then I'm concluding this session. So first of all, I'm very much thankful to the Dr. Lynn Denny for delivering a very wonderful and provoking talk on especially electrochemical research endeavors. She has highlighted many aspects, electrochemical research endeavors, then selection of journals, then few aspects related to research ethics. So really, okay, it is a wonderful talk. Definitely, okay, it would be very helpful to all the participants. So on behalf of organizing committee, once again, I am thankful to you. And see, okay, my collaborator. Recently, we have been published one paper in Nature and Scientific Report, and most of the work is related to the serotonin sensing. And in that, though we have synthesized different multifunctional the nanocomposites, but the applicative part is completed at the University of Strathclyde by Dr. Lynn Denny. So again, okay, we are thankful to Dr. Lynn Denny, and I am also thankful to Professor Anil Ghule for sharing this session. So at the end, I am thankful to the speaker, then chairman of this session, and all the participants. Okay, thanks to you all. Thank you so much. Bye bye. See you.
Thank you.
Okay.